Hello everybody. Today we're going to be talking about lab safety. And in particular we're talking about lab safety in the microbiology lab. So the first thing that we need to discuss is proper apparel when you're in the lab. So always have a lab coat on. You always have to have closed-toed shoes. And if you have longer hair, that needs to be tied back so that it's not in your face. The reason for this is that your hair could potentially A, be set on fire when you light a Bunsen burner, but B, you may actually get your hair in the microbes, and I don't think you want bacteria sitting at the end of your hair. One of the first things when you come into the micro lab that you have to do is clean your bench. Okay? So when you clean your bench, you're going to take some kind of a cleaner, and in this case we're using Vespine. You just squirt it on the surface of the bench, and then you take a paper towel and wipe it off. This needs to be done before you start and after you are finished for the day. And again, we have to do this to reduce the amount of bacteria that's on the surface. We don't want to contaminate you, and we also don't want to contaminate the samples that you're going to be working with. The second thing you want to do before you start your experiment is you want to wash your hands. Again, good hand washing is going to eliminate or reduce microbes being transferred from you to your specimen, or your specimen to you and to someone else. Again, hand washing, as with cleaning the benches, needs to be done before you start and when you are finished. Now in this lab, in addition to working with chemicals, we are going to be working with microbes. And there are certain types of microbes that we are working with in this class that you should be aware of. They are biosafety level 1 and 2. What does that mean? What that means is that if you use the proper safety precautions and you follow those precautions, these are low-risk microbes. They won't cause an infection. So you have to make sure you're adhering to all the safety precautions that are in this particular laboratory. So things you don't want to do: you don't want to open a tube, sniff it. You don't want to stick your fingers in it. You have to make sure that you use the proper implements and instruments when you are culturing things. This is a probe that we'll be using for some things later on. You should also make sure that when you're working with chemicals, again, that you are very cautious, that you find out from your instructor or look at safety sheets that are provided to you to find out how dangerous those chemicals might be. So some of the things also for any lab that you are going to be working in that you cannot do: number one, you can't eat. And especially in this lab, you don't want to eat any food. You can't drink anything in the lab. Again, if you're like me, I don't trust anybody. So you don't want to compromise your safety by bringing a drink in, putting it down, and someone else spilled something like bacteria, then you pick it up and you drink it. Also, you shouldn't be smoking in a lab. You should not be applying makeup in a lab. Don't stick your fingers in your mouth in a lab, etc. These are all just general good safety practices that you should always follow. So when it comes to doing some of the experiments and in some further videos, we're going to be talking about what's called sterile technique. So you need to use a Bunsen burner. So one of the things that you're going to want to do when you use a Bunsen burner is you just move the burner to a place that's very easy for you to access. Okay? You do not want to have too many things crowded around it, but have your samples relatively close to it. Have this done before you actually light your Bunsen burner. so you're not reaching over a flame. Before you turn the gas on, you want to make sure you've got sparks coming out of your striker. Once you're convinced that all of this is in a good spot, then it's time to turn your gas on. So you turn the gas jet on. You move the striker close to the Bunsen burner, and there you have a flame. You light it. You'll do all of your work then by this flame, because we need to do things aseptically, which we'll talk about later in another video. You work around the flame. When you are finished, make sure that you turn the gas off and that you just move your Bunsen burner back to a starting position. Once you're done with that, just take your Bunsen burner, put it back where it was before, and life is good. Okay. Now we've gone through some general things here. Now, what are you going to do with the bacteria after you've worked with it? What do you do with chemicals when you're done with them, if they're waste? So, first let's talk about chemicals. Chemical waste has to go in some kind of designated bottle that tells you this is chemical waste. You have to label on there what it is that you put in there and when you put it in there. When it comes to biohazardous waste, there are different ways to dispose of it. If you have something solid, such as a Petri dish, you can take a look at this plate a little closer. There's a big blob of mold on it. This has to go in a biohazard bag. If you have something like this, which is liquid in nature, these are tubes. Okay? These have bacteria in them. You're done with them. They need to be in a rack, and they need to go in a designated area to be autoclaved. Autoclave means sterilized. So you take this, make sure that it goes in the proper area. The last thing of a biological nature that you may be using would be some sharp instrument, like a needle, maybe a glass slide that you put bacteria on. Those need to go into a sharps container. What happens if you have an accident? What happens, let's say, if you spill something on yourself? Well, the labs at SPC are equipped with a shower. They're also equipped with an eye wash station. So you'd want to locate where those are in the laboratory that you're in and make sure that you find out exactly how to use them. Usually there's a chain to pull, a button to push, so that you can rinse yourself off should something like that happen. Also, if you cut yourself, you should have a safety first aid kit in your lab. Find out from your instructor where that is. And you need to make sure any cuts on your fingers, on your hands, are bandaged up, covered up, or you may want to wear gloves even over that so that you do not infect yourself with anything that's in the micro lab. Lastly, one of the things in this lab that you have to be aware of is that you're going to be working with antibiotics. You will be working with some chemicals that some individuals have allergies to. So, that being the case, please make sure that you let your instructor know if you have allergies, if you have any kind of medical condition, and always make them aware if there's any kind of an emergency that happens in the lab so that they can take care of it right away and that they ensure the safety of everybody else in the lab. If you follow these few steps that I've told you, adhere to these different kinds of things that we've discussed here today, you'll be successful, you'll be safe, and your lab should work out very well.